Hello, my name is David Sexton, and I want to welcome you to my session on Biogen's UK Biobank Data Challenge. I am the Senior Director of Genome Technology and Informatics at Biogen, and I'm joined today by Frank Notaft, who is the Technical Director in the Health and Life Sciences space for Databricks. And today I'm going to be talking about, in three different sections here, about what the UK Biobank is and what Biogen hopes to achieve with it. Frank is going to talk about the part of the informatics solution for analyzing this data that Databricks helped us with. And then in the final section, I'll talk about how we extended the solution from the UK Biobank to Biogen's own sequencing platform. So what is the UK Biobank and how will Biogen use it? Well, the UK Biobank is a long-term study in the UK of 500,000 British citizens in the population. It covers about 30 years of data with participants who are aged between 40 and 69 years old that were recruited between 2006 and 2010. And their long-term care and health records are followed over a long term, about 30 years. Most of this data is coming from the National Health Services, including all of the information about their healthcare records and demographic information. This information was collected from 22 different centers across the UK, and 500,000 volunteers were actually recruited across all of those centers. They were all consented to be part of this study and were willing to donate various samples to the study. So this is one of the largest and most detailed population studies that's ever been undertaken on the scale of similar studies that were done in Iceland and that are just now being started in the United States. So what data is actually collected by the UK Biobank? So in addition to samples that are being collected, demographic information is being collected, plasma biomarkers is being collected, MRIs are being collected and made available along with neurobiology information. Physical measures are being collected, including accelerometer data from devices that the participant wears. Cognitive assessments, the DNA is being used for genome-wide association studies, and then medical record information is being collected as well, including ICD-9 and ICD-10 codes. So just looking at some of the demographic information that's being collected, as you can see here, health-related outcomes are being collected. So what the outcome of the treatment of the patient was, whether that patient has died, any sort of cancer diagnoses, any inpatient information that would come from the medical record, and any early life factors, things like birth weight or anything else that would be present in the electronic medical record. There's socio-demographic information that is being collected, so information about the household, employment, any education, ethnicity information is being collected as part of the study itself. And this demographic information is typically being collected in questionnaires, and there are many questionnaires that are given to the patient over the course of the time of the study. Health and medical history information is being collected, so information about breathing or pain or any operations that the individual may have had, including any self-reported illnesses. And then there's quite a lot of phenotypic information about their lifestyle, so levels of physical activity, sleep, smoking, alcohol use, any family history information, and then mental health results as well. So Biogen was part of the Life Sciences Genetics Consortia, which is a large pre-competitive consortia that was set up in order to sequence all 500,000 of the UKBB participants. There were 8 companies who were involved with this: Regeneron, AbbVie, Pfizer, Alnylam, Biogen, AstraZeneca, Bristol Myers Squibb, and Takeda. as part of the original program, Regeneron and GlaxoSmithKline sequenced the first 50,000 patients, and all of that data is now in the public sphere, and any scientist can make use of that data right now. All 500,000 participants are to be sequenced by the second quarter of 2020. We're coming up on the end of the second quarter. We've been slowed down by the COVID-19 pandemic that is currently going on, but we hope to complete all 500,000 participants by the end of the year. As part of this consortia, all of the industry partners will have exclusivity to the data for 1 year after the completion of each tranche of data. So the tranches are 100,000 exomes at a time, usually delivered on a quarterly basis. So you will see a rolling release of the full 500,000 exomes over the course of approximately a year to a year and a half, depending on when we can complete the final sequencing post-pandemic. One of the key things that we're looking at here is genotyping and certainly sequencing information, and are quite interested in any associations that we would see in the genome to diseases that we have interest in. We currently have 300,000 exomes in-house, and we've been analyzing those on a rolling basis as we get more samples. So some key discoveries that have come from the UK Biobank over the course of the last 8 years are that there have been over 500 peer-reviewed publications at this point, all of which are improving our understanding of genetic and epidemiological factors that influence the disease, certainly that Biogen cares about, which are neurological diseases. So things you can think of here are depression, intelligence information, insomnia, addictions, and other brain illnesses, but certainly across other disease areas as well. There has been quite a lot that has been published from the UK Biobank over that time. And in the very short time that Biogen has had access to the data, we're already in the process of publishing a paper around rare protein truncating variants and their effects on human lifespan. So how is Biogen actually leveraging the UK Biobank data? So the first thing that we wanted to do with the UK Biobank data is to use the human genetic evidence that we find there to rank our drug portfolio. So we have found that over time that our drug portfolio, if we have human evidence for the disease that we're looking at, and we have genetic information about that, that we're able to rank our drug portfolio and actually get to market quicker with drugs over time. So we spent, with the first 300,000 samples, we spent the last 6 months going through and ranking our entire drug portfolio, which is about 60 different targets across 8 diseases. And we now have in the lab started to go through and work on the new ranking of our targets, where we've now moved around in our portfolio quite a few of our drug programs based on this evidence. We've used it to find new gene targets, so things that we may not have been looking at across our portfolio, looking at new genes that have now shown up in the genome-wide association study, and we're now testing compounds and molecules against those new targets to see what we can bring into our portfolio. And then I think a key portion of this is just understanding the neurological disease biology, and seeing if we can measure some of the outcomes from Alzheimer's disease, Parkinson's disease, and other dementias based on genetic evidence. So obviously, with bringing in this large bolus of data, Biogen needed to have an informatic solution that could be matched to handling data at this scale. So we had partnered obviously with Databricks, but also with another company called DNA Nexus, and coming up with a software as a solutions stack that sat on top of AWS that allowed us to analyze this data at scale. So some of the challenges that we had in our own data infrastructure bringing in the UK Biobank data was that we did not have enough storage capacity in our data center. And while we had approximately 8 petabytes of storage in our data center, bringing in another petabyte would have required us to spend $1 million purchasing new disks to install there. It also would have required us to purchase new switches and to purchase new compute to handle that new storage. We also found that we had network problems. So again, we would have had to purchase new switches, but also we would have been required to upgrade our internet connections and our connectivity to the sources of the companies who were providing the data. We also, in 2018, at the time when we signed the contract for the UK Biobank, we're having issues with our parallel computing infrastructure with crashes in our high performance computing system that required us to rethink how we were going to analyze this data. So really what we needed was a new data paradigm for Biogen. We needed to think about petabyte scale infrastructure, which is something that Biogen had not done previously. We'd never worked with data at this scale. We needed to be able to store and visualize genomic data. Again, we had never dealt with genomic data at this scale, and it required a whole new suite of tools for us to be able to interact with it. We needed to be able to analyze this data at scale. Again, we did not have the parallel computing capacity or really the software tools to deal with analyzing this data at that scale. And we also needed to be cloud first. This was something that that Biogen's IT department required, that we would not purchase new on-premises compute infrastructure, but that we should use the cloud wherever possible. So the solution that we came up with for dealing with this large new bolus of data was that we needed a secure solution to handle truly big data, which led us to Amazon. We had already had a virtual private cloud set up with Amazon, so it made sense for us to use that infrastructure, including obviously S3 storage, elastic storage, where we could put any size data, multiple petabytes of data in place in S3, and elastic compute on that data using Amazon EC2. And then for analyzing that data, one of the things that we wanted to do was to make it easy for our data scientists to set up their environment, their compute environment, and their analysis environment, and to be able to easily visualize the data and use common tools such as R or Python that they were used to using for analyzing the data in the cloud. So the architecture of this new infrastructure was to set up a new method for transferring data. Regeneron Genetics Center was the group that was actually sequencing all 500,000 UK Biobank samples. They were then sending the data to DNA Nexus, who were doing the primary and secondary analysis of the data to create CRAMs and VCF files. And then we were receiving that data from DNA Nexus in order to do tertiary analysis and trying to assign biological meaning to the data that we were actually getting. So all of this, because all three groups were already using Amazon infrastructure, it was very easy to move data via bucket to each group who had across virtual private cloud, so that each group could actually use this data. And as you can see on the right, the way that we had the architecture set up was that in the customer environment inside of our VPC, all of the data stayed inside of our VPC, all of the computing stayed inside of our VPC, and we worked with Databricks to create an orchestration layer that would create clusters using EC2 and allow us to compute on that data inside of our own VPC using the tools that the data scientists were used to using. And part of what we were very excited about with Databricks was using their genomics runtime. And Frank will now talk about the genomics runtime.
Excellent. Thank you very much for the introduction, David. So I'm Frank Notaf, Technical Director for Healthcare and Life Sciences at Databricks. And for those of you who aren't familiar with Databricks, we're the company co-founded by the creators of Apache Spark. So we started in 2013 when our team of co-founders, 4 grad students, 2 faculty at the University of California, Berkeley. They took the open source Apache Spark project and built a startup around it, whose goal was to put a platform together that would allow people to use data, data engineering tools as well as data science tools together in an easy way. So we actually provide a platform that automates the orchestration of Apache Spark in your AWS account. So we make it easy to set up these clusters in your account with minimal setup. We then provide a highly optimized Spark layer on top of that with a managed notebook style experience that makes it easy for people to spin up workloads and collaborate across them. Well, this technology can all be used very broadly across any industry. We started in 2017 going ahead and spinning up a healthcare and life sciences team. And in that, we've actually gone ahead and built out a product in the genomic space. We're one of AWS's life sciences competency partners. We've been very fortunate in the space. We have a very large practice in pharma space, obviously working with Biogen, but also working with another 7 of the top 10 pharmas. We work with 3 Fortune 10 healthcare companies, as well as major governmental organizations across the US, Europe, as well as Australia. When we look at the work that we're doing in genomics, and I'll dive into what we're doing in the space, we see applications all across the board. Obviously we're doing a lot of work in the drug discovery and target prioritization space in pharma. We've had a number of interesting workloads in the diagnostic space where we're working with large genomic data to identify biomarkers. We're working with large genomic data to build novel machine learning models that people can leverage. And we've started to see a good pivot into the healthcare space where organizations like Sanford Health are doing biobank scale sequencing in the context of a medical facility. They're using this to hopefully guide treatment to drive better outcomes for their patients. Next slide, please. So with the Databricks platform, what we've gone ahead and we've done is we've introduced a runtime. So this is kind of an optimized set of software installed around Apache Spark that is optimized for genomics. Again, this runs directly in your AWS VPC. We take data from your S3 account, your BAM files, your VCFs, CRAMs, FASTQs, and so on, and we provide pre-built steps that help you accelerate your data processing. First thing that we've done in the pipeline space is we've gone ahead and we've taken the GATK4's best practice pipelines in the germline and in the mutation calling space, and we've highly accelerated them. So you actually get about a 4x performance improvement relative to running the GATK on your own side. This will do things like drop your EC2 spend for these pipelines from $12 down to $4. Well, we also drop the time that it takes to process a single sample from 35 hours on a single EC2 node to under 40 minutes when running on a scaled out Spark cluster using Databricks. We've then gone ahead and done a lot of work downstream to make it easy to blend samples together into cohorts and parallelize tools out at large scale to run major tertiary analyses. So we've actually done a re-implementation of the GATK's joint genotyping algorithm to scale up to large cohorts in a managed offering. And then we've taken popular open source tools like Hail, Atom, and we've actually introduced the open source tool Project Glow, which is a collaboration jointly between us and the Regeneron Genetics Center, to really go ahead and push these tools, push these analyses to the next level of scale. When we finally see people get down to ML, the platform and its capabilities around reproducibility make it very easy to run large-scale analyses in a reproducible manner. For instance, one of the big libraries that our organization inside of Databricks has developed, us as a company, is MLflow, which is a next generation library that allows you to store machine learning experiments in a reproducible and shareable way. So when you go through, when you run your GWAS, when you build a new machine learning model that identifies rare variants, you can push all the code, all the parameters in the model that you've actually trained into MLflow, so you can easily share it and reproduce the full chain of lineage that's involved in this experiment. Next slide, please. At Biogen, we've been fortunate to work both across the secondary as well as the tertiary space. And while we'll talk a lot about the tertiary space today, on the whole genome side, we've been able to go ahead and help them with a couple of projects where they've had multi-thousand sample whole genome call sets that they've needed to take together, reprocess, and generate a cohort from that they can then use for tertiary analysis. There's a couple of big things that we've been able to do here. One, if we think about how complex it is to set up the GATK in the right way. I know back when I was working in the open source space on the Toil workflow runner, we were trying to set up GATK 3.5 and it took us 6 months to get it set up. Here we were able to set up the GATK in about 5 minutes. It comes as a prepackaged pipeline. You just install the notebook, you reuse the Databricks Genomics runtime, and it's ready to go. When we then moved down to the point where we had these GVCFs for each sample, with our joint genotyping pipeline, we're able to go ahead and run joint genotyping very rapidly under a day on a large cohort and get samples that we could, that were immediately ready for tertiary analysis. The way that we actually store this data is, we can go ahead and write out a VCF that people can use, but we actually natively store it in Delta Lake, which is an open source columnar file format that we've developed at Databricks and open sourced. And we use the project GLOW schema so that you can go directly from that kind of project VCF style data set and start running GWASs, start running variant annotation very rapidly. Next slide, please. What we've then done on the UK Biobank data is we've helped the team at Biogen build out a reproducible GWAS pipeline. They're mostly using the Hail tool, which is bundled into our genomics runtime, and we've actually helped to bundle up some of the common variant annotation functions that people use. So, for instance, one of the first things they wanted to do was annotate against a variety of databases. Annotate for loss of function variants, annotate with with kind of custom plugins. Previously, this took about 2 weeks to do 700,000 variants. When we scaled it up to a larger call set with 2 million variants, it took about 15 minutes, and this allowed us to ultimately scale up to annotating the whole UK Biobank with its 90 million variants post-imputation. When we then get to the GWAS space, we've been able to help them run these GWAS ultra quick. I think the first GWAS we were able to get all the way from loading the raw data in, quality controlling it, and making a first pass GWAS in under 2 hours. And ultimately, this has allowed them to build out a rapidly queryable database of genotype and phenotype associations. So I'll turn back over to David Sexton, who will talk a bit more about the work that the Biogen team has been doing with this data.
Thank you, Frank, for that detailed information. So after working with Databricks and with Amazon Web Services to set up this infrastructure for the UK Biobank sequencing, we decided to extend the platform to our own sequencing that we do at Biogen, both for research and for clinical samples. So Biogen has a large sequencing infrastructure of our own using instruments from Illumina, PacBio, Oxford Nanopore, and others to sequence our internal research data samples and our clinical trial samples as well. So we have the capability to obviously to produce data from those instruments for processing and detection of any events, for filtering and review and validation of the sequencing that we're doing for our individual internal projects, for annotating and predicting any variation that we see in the samples that we're sequencing, and then for interpreting and creating reports for any significant variants. And then we can apply this in the translational medicine space to any sort of clinical application that our clinical trial teams would want to use. So really the ultimate goal is to get to data-driven drug discovery. And this is a new capability that Biogen has not had before. And this is something that has been key in putting together our current drug portfolio. And so this is, again, another picture of a similar diagram that I gave earlier in the talk. But essentially what has happened is we've replaced the Regeneron data center with Biogen, and we're now using the same infrastructure that we were using for UK Biobank. So now we're able to sequence our data, have the files go to DNA Nexus for initial primary and secondary analyses, and then we bring them into Databricks for our tertiary analysis as well. So to wrap up, I just wanted to talk about Biogen's challenge again with the UK Biobank data. Here we were bringing in 500,000 UK Biobank whole exomes, which is a little bit more than 1 petabyte of data. Our infrastructure was not ready for this data, and we had a very short time frame to get ready, approximately 6 months from the time of signing the contract to being prepared for the first sequencing data to come in. We had been instructed by our IT team to have a cloud-first policy, and that we should use our Amazon cloud as much as possible. And then we also had a request from our research group that we extend this to all of our sequencing work. So what was our success here? We've actually repositioned our drug portfolio with genomic evidence, and we have promoted some drugs and demoted others in our portfolio based on the information that we have from the UK Biobank. We found a couple of new drug targets that are interesting that we are now in the process of following up. And we're teasing apart the biology of neurodegenerative disease. And I think this is something that's very important beyond just Biogen, that we have a better understanding of degenerative disease, specifically around Alzheimer's and Parkinson's disease. So with that, I will thank everyone who's been part of this project. Especially want to thank Frank in the work that Databricks has done, and I want to thank the organizers and Amazon for giving me the opportunity to talk.